have the pleasure of having in our series two amazing neuroscientists from the University of Washington University Medical School, Dr. Meaghan Creed and Dr. Tamara Hershey. And then I'll introduce both of them, and then we will have Dr. Creed to give her seminar, her scientific seminar, followed by the recruitment component that will be led by both Dr. Creed and Dr. Hershey. Let's start with Dr. Meaghan Creed, who is assistant professor at the Departments of Anesthesiology, Psychiatry, and Neuroscience at the Washington University Pain Center at the School of Medicine. Dr. Creed earned her bachelor's degree in science and PhD from the University of Toronto in Canada. And during her graduate work, she studied deep brain stimulation, something that there's some very much interest from myself since I did my master's on intracranial self-stimulations, which is more or less the same model for studying addiction, and applied to the basal ganglia in models of movement disorders. For postdoctoral training, she moved to the University of Geneva in Switzerland, where she used optogenetic tools and electrophysiology to understand how addictive drugs rewire synapses in the brain reward system. And in order to treat any psychiatric condition, we must first understand its underlying pathology. Therefore, Dr. Creed's research seeks to integrate a variety of approaches to understand how plasticity in the brain's reward system underlies maladaptive reward-guided behavior in models of addiction and chronic pain. And her ultimate goal is to apply treatments like deep brain stimulation to modulate neurocircuit function and to treat behavioral symptoms of chronic pain and substance use disorders. In addition, this afternoon we have Dr. Tamara Hershey, who is the James McDonald Professor of Cognitive Neuroscience, and she's the director of the McDonald Center for Systems Neuroscience and co-director of the Neuroscience Phd Program, and of course, a highly regarded scientist in the field of imaging. So both of them will be talking to you, so it's our pleasure to have you both. And then I will allow them to have Dr. Creed upload her presentation and we can enjoy her talk. Thank you, Dr. Creed and Dr. Hershey, for being here. You're still muted. Oh, yeah.
Thank you.
Sure.
Okay. And so I did one practice with this. Let's hope we go. Okay, and so I can't see anybody in this presenter view, so if people have questions, feel free to jump in and interrupt. I think that's okay?
Yes, please. Thank you.
All right, and just one housekeeping item. I'm sure a lot of us have been up late over the night, following the news closely, so a little bit of grace as we go through this. But basically, I'm really excited to be here and to tell you about the work that I've been doing in my lab at the Washington University Pain Center. And it really builds on lab work that I did out of my postdoc lab at the University of Geneva. But the main principle here is that my work seeks to leverage insights about synaptic plasticity, or how the neurons in the brain communicate, and parlay this into ways that we can treat disorders of reward processing. So what do I mean by a disorder of reward processing? So this is the general approach that we take to studying disorders of reward processing. And the case study that I'm going to tell you about to start off today is really a vignette looking at cocaine abuse or cocaine addiction. So this is the general schematic that I follow in my research. The first step is to determine which neural circuits are causally implicated in behavioral symptoms or maladaptive.
I'm sorry, we don't see your presentation.
No?
No.
Okay.
You need to share.
Yes. Are you seeing it now?
It's coming. It's coming. Right. So now
Okay.
just have it on slideshow.
Okay. Okay. Are you able to?
No, it's still on a different, you know, when you can see all the slides on your left. So it's not.
Okay.
If you go down to where the projector on the bottom of the PowerPoint, maybe that will help.
Okay.
There's this icon. That's exactly. Let's see if that works. Nope.
So it's giving me a list. I emailed you the slides as well, if that's easier.
Yeah, I can go ahead and do that. No problem. Is that good for you?
Yes.
Okay, we'll do that. It's coming.
Okay.
You just sent one, a new version, right?
Correct. So.
No worries, we'll get it. It's just that it's because it's checking if it's a virus or something. But I already downloaded. It's just a matter of a couple minutes.
Okay. There. All right. Is it the format is strict? It makes sense to keep working on this instead of doing Tammy's part now and coming back to me.
Just a couple minutes, I think, will be fine. Let me just go back to.
Wouldn't be a modern day talk if we didn't have some technical difficulties, right?
Exactly. Exactly. It's taking a while to open. I don't know why, but we'll get it.
Okay. All right. And now I'm trying once more. So it's, allow Google Meet to see your slides.
Okay.
I'm clicking on my PowerPoint show. I'm clicking allow.
Okay. Can everybody see it? Not yet. Okay. Well, I have it now. No worries. It's coming up pretty. One more minute. So how's the weather in St. Louis?
It's great. It's beautiful.
Oh, wonderful. I'm glad.
At least we have that.
Yes, I know. I feel for you. I didn't sleep last night, actually. Well,
yeah.
Here we go. Okay, I have it, Megan. No worries. So let me share my screen. Okay, I'm going here. I am window, and I see it. This is it, right? Yes?
That's correct. Yes.
Okay.
All right. So
I'm gonna, hold on. I just lost it. Where is it now? You see, everyone sees it? Wait, everyone sees it now?
Yes.
Okay, I'm going to, there. Is that good? Everybody can see it?
everyone can.
Yeah.
Yes.
Wonderful.
Okay. So
So
all
now,
right. So this
just tell me.
Okay, so of course, I've added a lot of animations that need to have this clicking, so we'll just see how this goes. Might break up my flow a little bit, but this is good.
Okay.
So I'm excited to talk to you about the work that my lab does, looking at rewiring the brain's reward system in order to treat disorders of reward processing. And I'm going to start by telling you about the process that we follow, a vignette applying this process to the case of cocaine addiction, and then moving on to some of the work that we've done more recently from my lab, extending this into models of opioid abuse and chronic pain. So especially with the animations being a little bit choppy, I think that's great. It will give us time. If you guys are having questions, just jump in and please interrupt me and ask questions for clarification as we go along. Great, so next.
Hold on. Next, yes.
All right. So this is the general process or system that I follow in my lab and when I'm thinking about science and designing experiments. The first step is to determine how specific neural circuits control specific behaviors, especially how those behaviors go awry in psychiatric disease. The next step is to understand disease-driven adaptations within the circuitry. And finally, to understand how we can modulate or change function of these circuits in order to treat behavioral symptoms of psychiatric disease. And as I mentioned, I'm going to tell you a little bit about how this works using a case study of cocaine addiction. Next. Next. So addiction, as it's classically defined, is the repeated compulsive use of a drug or a substance despite significant negative consequences. And one of the main issues with treating patients with addiction in the clinic long-term is that addiction is a chronic relapsing disorder. And that means that even after a patient has been able to stay abstinent for weeks or even months without taking any of the drug, exposure to cues or to environments that are associated with the drug can still evoke these powerful feelings of drug craving that can then lead to relapse. So this idea of what's changing in the brain, what plasticity is underlying this persistent behavior long after the drug is out of the system. Next. And the circuitry that we're going to focus on for understanding this plasticity is this classic mesolimbic reward circuitry. I've shown here, this is a schematic in a human, focusing specifically on the prefrontal cortex, the medial prefrontal cortex, and its inputs to the nucleus accumbens, or the NAC, I might call it in shorthand. So the nucleus accumbens, or the NAC, is this brain area that synthesizes excitatory inputs from a variety of different brain areas, including the cortical areas for executive information, hippocampus for memory-related areas, amygdala for social and highly charged valence-related information. And it synthesizes these inputs and then transmits them downstream in order to guide reward-related behavior. And next. And we use rodent models to study the circuitry, but the circuitry that I just simplified and outlined is really homologous in the rodent. We also have the accumbens that's integrating these excitatory inputs from the medial prefrontal cortex, MPFC, hippocampus, and amygdala, and then transmitting it downstream. And as will become important for later in the talk, a lot of these areas receive dopamine. Next. So dopamine from the ventral tegmental area innervates all of these targets, which will become important in just a second. Next. Next. All right, so we use tools called optogenetics to selectively manipulate these different circuits. So here we're showing just a schematic of the experimental paradigm that we're going to use for this first vignette, really focusing on excitatory inputs from the medial prefrontal cortex into the nucleus accumbens. Next. And when I say channel rhodopsin, this is what you're seeing here. This is a protein that was originally isolated from bacteria. It's similar to proteins that are located in your retina, where they're light activated. So if you hit these proteins with light, they open up and they allow ions to flux in. So we can express these in specific neurons in the brain, these light activated proteins. And then when the neurons are expressing these proteins, whenever light activates those neurons, the receptors open, you have excitatory input ions coming into the cell, and they set off these action potentials. So you can actually use light to control the firing of neurons and to selectively isolate the neurons you want to study, because you can selectively express these proteins in specific areas of the brain. So here what you're seeing in the bottom left panel is us injecting a green virus, which is our channel rhodopsin or our excitatory opsin. We're injecting it into the medial prefrontal cortex, which is what you're seeing in the left-hand panel. And then the image with the blue frame is the image of the cell bodies that are infected. The virus that expresses this protein is going to go all over the surface of those neurons, sort of like a breaded chicken cutlet, where you have all of the neurons coated in this protein. And this includes their processes that extend down into the ventral striatum or the nucleus accumbens, which is what you're seeing with those two optic fibers going into the nucleus accumbens. And we can use the light, use optic fibers implanted into the brain to stimulate the terminal fields of these neurons, so we can selectively isolate the effects of these cortical inputs into the nucleus accumbens and determine the effects that activation of this pathway has on behavior. Was that much clear? It's a little bit, I'm trying to be really explicit without a pointer here. Any questions? All right, next. Okay, next. Okay, and so then the behavior that we're going to look at is, as I mentioned with the, when we're modeling addiction, one of the hallmarks is that the behaviors that we want to use to model addiction, they have to appear only after repeated drug exposures, and then they have to persist, they have to outlast the presence of drug in the brain. And we use a really simple assay of drug-associated memory, which is locomotor sensitization. So basically what you're seeing in this graph is the open circle. When you give a single injection of cocaine, mice are activated. Cocaine is a psychomotor stimulant. The mice will run around and be very activated, and we can quantify how much they move. However, the interesting point is that if you give that identical injection of cocaine after a week of withdrawal, that effect is really enhanced. So there's this psychomotor sensitization in response to an identical injection of cocaine where the mice are even more activated. And this is dependent on the environment. So it only happens if the mice receive cocaine in the same environment where they received their first injection. And it's thought to reflect a motor readout of a drug-associated memory. And on the next slide, we have a video of what exactly that looks like. So I think to play it, you have to click on the frame.
We'll try, but I don't see anything.
Yeah, it's on the, yeah.
And now?
No, okay, so the video skipped, but we can talk about that later, but this is okay. So we know that the cocaine induces this behavioral activation, this behavioral sensitization with repeated injections. And we know that this is related to the strength of that connection between the cortex, the medial prefrontal cortex, and the accumbens. And we can assay the strength of that connection directly using techniques like patch clamp electrophysiology. So this is actually taken from my lab. That's me doing some patch clamp electrophysiology to measure the strength of this synapse. So it's the same preparation that I described before. We have our channel rhodopsin infected in the medial prefrontal cortex. But instead of stimulating over the accumbens in vivo in an awake behaving mouse, we take slices of the brain, and we can put the slices under the microscope and visualize them. Next. And what you're seeing here is a glass pipette, which is containing an electrode that's connected to an amplifier that's going to allow us to measure what's happening inside the cell. So under that microscope, we're approaching the cell with our glass pipette. Next. And we eventually use the pipette, which is a very fine hollow glass tube. This is an individual neuron, to make a seal with the pipette. And then we apply negative pressure, breaking open the cell membrane only in that area of the pipette so that our electrode essentially is in the middle of the cell. We can eavesdrop on what the cell is doing and listen to it electrically. Next. So this is an example recorded on my rig, just listening to spontaneous action potentials that a cell is firing. Next. Or what we can do to integrate this with our optogenetics. Here you're seeing that green neuron, which is expressing our green virus, and it has the channel rhodopsin virus in blue. So when we activate those green terminals with light, they're going to fire action potentials and spit out neurotransmitter. That neurotransmitter is going to cross the synaptic cleft. And then if we're recording from the purple cell, which expresses receptors for that transmitter, we're going to see voltage changes that we can report based on the light activation. And this is an example of what that looks like. You're seeing those little blue pulses over the traces are the light pulses, and we see this inward electrical current that we are measuring the response of the postsynaptic cell. So is that much clear, the general idea of what we're measuring here?
Yes.
Okay, next. All right. And so this is a property of excitatory transmission onto the nucleus accumbens, that in a baseline state, most of that excitatory transmission is mediated by these classical or canonical AMPA receptors, which are shown in this diagram here. And I don't want to go too far into the weeds. Next. But following exposure to cocaine, we actually have a switch in the composition of those subunits. Next. Such that we have these GluA2 lacking or non-canonical AMPA receptors being inserted into the membrane. And so you can see in the two traces, the one from control and the one from cocaine, abbreviated as COC, C-O-C. I won't explain the biophysics of why this is the case, but basically when you see this pattern of less current fluxed at positive membrane potential, so this inward deflection shown in the blue trace, that's an electrophysiological signature of these GluA2 lacking or non-canonical AMPA receptors. But essentially what we're showing here, next. Essentially what we're showing here is that after cocaine, we have a switch where that excitatory transmission is no longer being mediated only by classical AMPA receptors. You also have these non-canonical AMPA receptors being expressed in the postsynaptic membrane. And we can read that out by measuring the AMPA transmission, like I'm showing in those representative traces. Next. So we basically have this situation where we know that cocaine strengthens or potentiates the cortical striatal projections. Next. And just a bit of background for anybody who's interested in the molecular mechanism of why that is. We know that, as I mentioned, the VTA, or the ventral tegmental area, sends dopamine projections to all of these brain areas downstream, the prefrontal cortex, accumbens, and ventral pallidum. But essentially what cocaine is doing by releasing this dopamine, when it hits the neurons within the nucleus accumbens, sets off this intracellular signaling cascade. Next. That ultimately leads to more of these non-canonical AMPA receptors being expressed at the cell membrane. So this is just a diagram that I made that shows my understanding of how this happens that you're welcome to look into. But the main takeaway here is that cocaine is potentiating the strength of these cortical synapses into the accumbens by inducing a functional LTP, a long-term potentiation by causing more of those receptors to be expressed at the membrane. And this is dopamine dependent, which will become important. Next. Okay, next. 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 So this is just confirming the model of this synaptic strengthening. Next. So our question then, if we have this drug adaptive behavior that emerges with repeated cocaine exposures, and we know that repeated cocaine exposure is increasing the strength of that synapse from the cortex to the accumbens, our question is then, can we reset this brain's plasticity and normalize or reverse drug adaptive behavior? Next. So how would we do that? And we know that from decades of classical pharmacology, one of the really efficient ways to remove these receptors from the synapse is by inducing a long-term depression, or an LTD, that's dependent on activation of these metabotropic glutamate receptors, or mGluRs. So as I mentioned, cocaine is strengthening that synapse. What we're trying to do here is depotentiate that synapse. And the reason this depotentiation works is that the glutamate that you're seeing being released from that presynaptic terminal in gray, it's releasing glutamate that's hitting the postsynaptic membrane, and it's spilling over and it's activating these extrasynaptic metabotropic glutamate receptors. And when those are activated, when you get enough glutamate efflux that those receptors are activated, you set off a signaling cascade that will ultimately dephosphorylate and internalize only those non-canonical AMPA receptors that typically aren't at the postsynaptic membrane. So it's a really targeted way of reversing that cocaine-evoked plasticity. Next. And so how do we achieve this in vivo? Next. We know that a really efficient way to do this is by using that channel rhodopsin. Here you're seeing that gray postsynaptic terminal expressing the channel rhodopsin. And we know that if we stimulate at a very specific frequency between 10 and 15 hertz, that we get just enough glutamate efflux that the glutamate is coming and spilling over and activating those metabotropic glutamate receptors without stimulating too much, that we're actually getting a further strengthening of the synapse. So we're really in this sweet spot of stimulation between 10 and 15 hertz. And we know that this is sufficient for inducing long-term depression or normalizing cocaine-evoked plasticity. Next. Next. And we can do this with optogenetics quite handily. We can do this in vivo using that same preparation I showed before, where we're injecting the virus into the medial prefrontal cortex, stimulating over the terminals in vivo. And we can do this in vivo. And if you look at the graph that I'm showing, in the control mice, I'll remind you, this is the main sensitization effect, where that open circle is the first control injection of cocaine. That open square is what a normal mouse does the second time it sees cocaine in that same environment. But if you look at the laser columns, the ones that are the solid blue circles, as we see the very first injection, there's no difference. But if we do this optogenetic depotentiation, if we stimulate this pathway in vivo, in the freely behaving mouse, if we play this pattern into the brain, 24 hours before we give them more cocaine, they act like mice that are seeing cocaine for the very first time. And this is because we know that at this very specific stimulation frequency, we're resetting that synapse that's important for the behavior. Next. So this is really great. We know with optogenetics, we can reverse synapses and reverse behavior and make mice that are seeing cocaine look like mice that are seeing cocaine for the very first time. So it should be great news, but there's a little bit of a problem. Next. And that's that optogenetics, we can't, if our ultimate goal is to treat addiction in the clinic, optogenetics can't readily be translated to humans. We can't immediately express viruses in human brains without significant difficulty and toxicity issues. It's also really hard to deliver light through areas of tissue. So it's limited clinical efficacy of what we can do with optogenetics. But something that I had done during my graduate work and something that's really promising and emerging as a treatment for many psychiatric diseases is deep brain stimulation, or DBS. And DBS is a surgical therapy whereby electrical current is implanted into electrodes are implanted into specific nuclei in the brain. Here in the picture, you're seeing electrodes implanted into the nucleus accumbens. We've done this in mice, but there's actually clinical trials where they've tried deep brain stimulation of the nucleus accumbens to treat addiction. Most of these have been done in Europe with mixed efficacy, and it's because we don't really know the mechanism. But our idea was, well, if we know that optogenetic stimulation at 10 to 15 hertz has this really robust, long-lasting effect on behavior, maybe we could do this with deep brain stimulation, which is something we can actually do in patients. Next. So we tried that exactly. Next. Next. So we tried stimulating the synapse, and we used the same range, 10 to 15 hertz, but we didn't see any reversal of the plasticity. Next. And I'm not showing it here, but we also didn't see any effects on behavior. So mice where we had applied the DBS, we really didn't see any, they behaved just like mice who had not had DBS. They still sensitized to cocaine, and their brains totally looked like mice that had received cocaine. We didn't move the needle on the synaptic change or the behavior. So it forced us to ask, why are the effects of optogenetic stimulation so different from those of DBS? Or as postdoc Meaghan would put it when I was doing these experiments, next, is why doesn't this work? So what exactly is different? But it did, next. So it did force us to take a step back and think about how these things are actually doing quite different things to the brain. So we see on the left with our channel rhodopsin stimulation, a lot of the power of that technique is that we're really accessing specific neural circuits. So we're only infecting and activating the neurons in the PFC that project to the accumbens. These are all glutamatergic neurons, and we're really selectively activating them to get this glutamate efflux. However, when we do the deep brain stimulation electrically, which is the middle panel, we're totally non-specific. So we're activating excitatory inputs from different brain areas. We're probably having effects on the local cell body, and we're activating other inputs that aren't necessarily glutamatergic. And if you remember, one of the ones that I told you about before was that dopamine, it projects to the nucleus accumbens, and electrical stimulation would be predicted to affect dopamine release as well. And if you remember, the way that cocaine initially induces this plasticity is by activating dopamine neurons or inducing dopamine release in the nucleus accumbens. Next. And if we drill down a little bit into the mechanistic level, these are the two signaling pathways I showed you before. That when you activate the D1 receptors, this is on the right side, the signaling cascade, not only does that D1 signaling increase the insertion of AMPA receptors into the membrane, it also phosphorylates and inactivates the enzymes in the mGluR1 signaling cascade. So ultimately what we think is happening with our electrical stimulation is that we're activating two opposing signaling cascades that are washing each other out, and we're not seeing any effect. Next. And we can test this directly on the slice. So what I'm showing you in the top is looking at measuring excitatory transmission from the prefrontal cortex onto the accumbens using patch clamp physiology. What you see, those first 5 points is the baseline, so the synaptic transmission at the baseline. Where it says 12 hertz is when we're applying 12 hertz stimulation with the channel rhodopsin. So that sweet spot between 10 and 15 hertz that we know gives us an LTD. And this is what we see, is that after that 12 hertz stimulation, that transmission is significantly depressed. It's only about 40% of what it was at baseline. But if we do the same 12 hertz stimulation, but with electrical stimulation, that's what you're seeing in the red open circles, we have no effect. So the 12 hertz electrical stimulation is not doing anything on its own. And this is consistent with what we see, the lack of the effect in vivo, the lack of the effect on cocaine sensitization. But if our hypothesis was correct, and that it's really these two competing signaling cascades, we'd expect that if we blocked the D1 receptor and applied the stimulation, then we might be expected to unmask or reveal that LTD that we see with the channel rhodopsin, because we're shutting off the dopamine signaling that's opposing and inactivating that long-term depression signaling. And we can test this in the slice. So next. So we repeated, here I'm showing you, we're repeating the exact same electrical stimulation, but this time, but this time we're repeating the exact same electrical stimulation, but in the presence of a dopamine D1 receptor antagonist or a receptor blocker. So we're washing on a drug. This drug, it's called SCH in the figure. It's actually a shearing compound that's clinically approved to treat Tourette syndrome. So it's something that's already put into human patients. And if we are doing this on the slice and we stimulate, we now, in the presence of the D1 blocker, we now see something like what we see with our optogenetics, where we get a sufficient or an effective decrease in the synaptic plasticity. Next. Next. So this is our model, that if we stimulate at 10 to 15 hertz, we get a nice, we essentially block, next, sorry. Yep, next. So repeating it, yep, next. Right, okay, great. So this is our model of our optogenetically inspired deep brain stimulation, where we're essentially stimulating in the presence of a dopamine blocker and seeing, and now we're unmasking that LTD and we're reversing our synaptic plasticity. Next. 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 And as I mentioned, we're doing this in vivo, so that's our model that we can, we think that we're inducing we're reversing the synaptic plasticity in slice. And now we can actually go in vivo and measure that. So in this experiment, which is one of the last slides I'll show you, we're stimulating with our optogenetically inspired DBS. So we're stimulating at a very low frequency in the presence of a D1 antagonist. And now, this is what, to remind you, this is what we saw before, where if we do the patch clamp physiology, we see this really characteristic hallmark electrophysiological signature of cocaine. Next. But if the mice have undergone this optogenically inspired deep brain stimulation before we record, their synapses actually look like synapses from mice that have never seen cocaine. Next. And then on a similar note with the behavior, what we're seeing is that if the mice have undergone the optogenetically inspired deep brain stimulation, or the OI-DBS, when you give them cocaine again after withdrawal, they're behaving like mice that are seeing cocaine for the first time. So we've also abolished this sensitized or drug-associated behavior. Next. So what I've shown you so far is that optogenetically inspired deep brain stimulation reverses cocaine-evoked plasticity and the sensitization behavior. Next. And I think for the sake of time, I can just summarize this one slide, and then I might pick one from the end to show you because it highlights work of a BP and DER student, but I think we'll call it there. But what we're doing in my lab now is really focusing, sorry, I should mention that protocol that I talked to you about in the first part of the talk it's now in non-human primate trials in Switzerland because the deep brain stimulation is something we already do in people, albeit sparingly. The drug that we propose to use is already FDA approved for people. So really proposing the novelty there is proposing this combination of a specific stimulation frequency and this drug ultimately to hopefully treat addiction or to manage symptoms of craving in patients with addiction and allow them to seek other means of treatment and allow them to sort of get relief from that craving symptom in order to combat their addiction and do the other work that's required to treat their addiction. Since starting my own lab, we've really shifted focus away from cocaine and more into the comorbid opioid use disorder and the intersection with chronic pain. And this is the one slide I want to show you because it was really striking when I put this together, that if you look at the amount of comorbidity between these different disorders, so CP is chronic pain, mood is mood disorders like depression or anxiety, and then SUD is substance use disorders. And I just want to draw your attention to the bottom left column. If you look at, of the patients that have a substance use disorder, over a quarter of those patients also have chronic pain. And then if you look at the top row, the right column, of the patients that have chronic pain, that are presenting with chronic pain as their common complaint or their initial complaint, more than a third of them also have a comorbid substance use disorder. So this is really striking when I learned these statistics. And what's even more striking in addition to the comorbidity or the degree of comorbidity is that if you look at the circuitry involved, for example, in human neuroimaging studies, a lot of those symptoms have been linked or implicated to metabolic changes or functional connectivity changes within the same reward network that I told you about before. So now in our lab, we're really focusing on understanding plasticity downstream, particularly in the ventral pallidum. So where the nucleus accumbens is what I told you about in the first story, but where is that information going? And one of the outputs is the ventral pallidum, which then links on back to the ventral tegmental area and to motor outputs. So the ventral pallidum is really this limbic motor interface, and this is where we're intervening, and we've discovered a lot of interesting things about that nucleus and how it changes in reward-guided behavior. And then I think if we can fast forward a couple slides, I'll just quickly highlight, we'll go through quite a few slides because I just want to show you the very last one, which was done by Maya Blewett in our lab. Here we go. So we'll go back one slide just to tell you a little bit about how we're modeling oxycodone use. So the model that we're using now to model oxycodone abuse is a really simple assay where we give mice the chance to drink oxycodone or their home cage drinking water. And my lab has developed these little devices that sit directly in the mouse home cage. And we just let the mouse choose whether it wants to drink oxycodone or whether it wants to drink water. And what you're seeing in the little GIF there is a video of a computational approach we have to scoring the biomechanics of the mouse face as they're consuming the oxycodone and looking for changes in their reward-seeking behavior as they develop a dependence and addiction. And in the very last, the next slide, next, I'll just point out that Maya Blewett, who was a BP and DER student for the last 2 summers in our lab, she did a lot to help us develop this device and validate it. She's now doing her PhD at University of North Carolina, and we're really proud of her. But just to say, this is one example of the sort of work that BP and DER students have done in our group, which we're really excited about. So I think for the sake of time, I will stop there and apologize again for the technical difficulties. But if we skip to the very last slide, oh, we're done. Never mind. It's okay. I will say that on the very last slide, I just have my web address where I've posted this talk already, as well as some of the methodological slides that have more information about any of the techniques that I've talked about. So if you have any questions about the methods or would like to get in touch, you can find all of those details on my website. And again, sincere apologies for the technical difficulties here.
Thank you, Meaghan. That was a wonderful talk, despite the difficulties. No worries. Open for questions from students. Let's see what we have in the chat. There's some. Okay. There's Carl, one of our graduate students at the medical school. He asked, I know you have discussed alterations in synaptic activity of this network. In your opinion, can alterations in excitability, such as modulation of ion channels, play a role in regulating induced excitability?
Oh my goodness, that's a really good question. And I'm kicking myself for going too slow through those slides. So on one of the last slides I was going to show you gets at exactly this question, albeit in the context of oxycodone addiction, which is exactly where we're focusing right now. But the short answer is yes. So I focused on the synaptic plasticity because we know that very specific information is carried between the medial prefrontal cortex and the nucleus accumbens, and there's a lot of behavioral pharmacology and classical literature implicating that pathway specifically in the behavioral sensitization that we're looking at. But absolutely, when we think of the more complicated effects of addiction, so things like the negative affect emerging after withdrawal and the long-term changes in excitability of these different pathways and how they interact, this is exactly the direction or exactly what our lab is focusing on. So changes in intrinsic excitability of different subpopulations within the ventral pallidum, which we know play a really important role in modulating the function of this network long term. And I'll cut myself off there before I go on and want to ramble on about it. But yeah, absolutely. If you'd like to talk more, I could talk your ear off about that. But I think that's a really, really important question, and absolutely.
Thank you. Any other questions from the audience? Let's see. Yes. This is one of our also undergraduates. Cielo says, considering that is also improving regular learning, do you consider your research on synaptic rewiring can be applied to educational policies to make visible how certain academic practices influence reward pathways?
So a couple of things, two points that I want to bring up there. The first is that this type of plasticity that we see is not really involved in general reward learning. So I agree totally the nucleus accumbens is involved in reward learning. This insertion of non-canonical AMPA receptors, this is not something you typically see. You see these receptors early in development when the synapses are being initially wired, for lack of a better word, but in a mouse, 2 to 3 weeks postnatally, these receptors are no longer there. And these receptors really only come back if the mice are exposed to drugs or some potent dopaminergic stimulation. You don't see it if you, for example, train the mice just to learn to lever press for sucrose. That's not sufficient to induce this plasticity. It's almost like cocaine is reopening this critical period of development where new synaptic connections and memories can be made much more easily. So in terms of how can it be applied to educational policies, I'm not sure. Maybe for some people, educational policies could be sufficient to reopen, to release enough dopamine. If somebody's really motivated to study or really interested in the subject matter, you can get sufficient dopamine to reactivate and drive insertion of these AMPA receptors. But I don't know. I will say, just bringing up your question, it reminds me of a talk I heard recently. They were using vagal nerve stimulation, which is known to increase dopamine efflux and norepinephrine efflux. And they were using this in dolphins to train them to sniff out landmines better. And they showed really convincing evidence that you could sort of mimic this surge of dopamine and norepinephrine and speed up how quickly dolphins could find landmines. This was a DoD funded grant I'm not supposed to be talking about, so let's not go into details, but all this to say that it is possible that people could harness this for improving learning, but I don't know whether your question about could you tell if learning has occurred based on the plasticity, I'm not sure that would be the case exactly. I hope that answered the question.
Any other questions? Well, I have a question. I'm very interested in what the combination of the DBS and the sharing compound in humans. So, is it you mentioned that in Europe they're considering like more toward a clinical trial to see if at what stage of addiction you would think that will be appropriate to start this kind of treatment? Are we talking about would you foresee, having an addict have the electrode in the pallidum? Is it where would it be first of all located the electrode for the DBS?
Right. So I think what we proposed would be what we'd proposed based on the data I showed you is the nucleus accumbens, although I have my own feelings about the pallidum long term. But yeah, so there's been a case, a series of, I think it was initially 12 patients, and then it recently expanded to 20 patients where they did do deep brain stimulation of the nucleus accumbens to treat alcoholism. It was Amsterdam and Germany were the two countries this was done in. And they applied deep brain stimulation. The main difference is that it was 130 hertz. They basically take what's used for Parkinson's disease because that's what's approved, and they apply it to the nucleus accumbens without any rationale as to why that would work. So you don't know the mechanism, which makes it impossible to optimize. And what we found was that it was really, really mixed effects, that patients did tend to report lower craving, but it was a weak effect. If you put them in a scanner with the DBS and showed them cues, you saw less accumbal reactivity to the cues. But it wasn't clear that this did anything for preventing long-term relapse. However, in terms of setting a precedent for what we think we could do, people are already putting electrodes in the nucleus accumbens, and the sharing compound is already something people take long-term. What I didn't mention is that this is a 10-minute protocol. So we give, the stimulation is only 10 minutes, and you only have to take the sharing compound once. And at least in mice, we've shown that mice behave normally and their synapses are still reset up to 3 weeks after that 10-minute intervention. So whether it's feasible, I think, is another, logistic, ethical, moral, political, financial consideration. But in terms of, it was really striking doing this work in Europe and then coming to the States and seeing the different attitudes towards it there, because in Switzerland, it was like, absolutely, why do you have to go to non-human primates? Why couldn't you do this right away? Whereas in the States, people are a little bit aghast at this idea. So I won't comment on the different attitudes there, but I think in terms of, there's already a precedent for what we're doing or what we're proposing, in that people have already gone through these procedures. And I think in the era of the opioid crisis, it's hard to argue that, there clearly are patients who really are needing novel and creative treatments.
Certainly. I just that I was a little bit interested in what would be the response, because there's so much data about I know a little bit about it, of animals self-stimulating the medial forebrain bundle, which is the axons that come from the VTA to the accumbens, and that's rewarding. So the animals will press a lever for that. And there's a whole literature on this matter. So having a deep brain stimulation in the accumbens, my reaction will be, well, then they will have a release of dopamine there. Of course, if you give the D1, like you mentioned, then you're blocking postsynaptically the response. So that I got that one. So okay.
Yeah. I think it's also stimulating in the terminals up in the accumbens is a lot less efficient than stimulating right in the bundle as well. But yeah, I agree. And we didn't look long term to see if the mice didn't seem to enjoy the DBS without the dopamine antagonist. But it's really hard to say if they would get addicted to their DBS or anything like that.
That is one thing that's why I was asking at what stage of the addiction we're talking about, because if it's early, if it's maintenance, or if it's when you're relapse, there's all sorts of, the brain changes throughout that cycle of addiction. So
yeah, and these changes I'm describing are only after protracted withdrawal or protracted abstinence. So it would be by definition in the late phase. So patients who have been able to get some amount of abstinence that are looking for relief from that craving type symptom. Right. To anthropomorphize, I'm dealing all in mice, obviously.
Yeah, well, this is wonderful. I'm glad I learned something today. I appreciate you sharing your data with us. So if there's no other questions, oh yeah, there's one from Christian. He says, how do you determine the observed effects are mediated by the D1 receptor? Could the other receptors be involved? D2, D3, D4, D5?
Yeah. So I think I didn't mention that the this plasticity that we're seeing, it's not on all of the accumbal neurons. It's really only the nucleus accumbens has populations that express D1 or D2. And we only see this plasticity in the neurons that express the D1 receptor. All the data I showed you was only from the neurons that express the D1 receptor. So that's one strong piece of evidence that it's D1 dependent. And then we did do a whole lot of control experiments that I wasn't going to show you that does show that it's a D1 antagonist specifically, and a D1 antagonist specifically infused into the accumbens also recapitulates this effect. We didn't see anything with the D2 antagonist. We just saw sedation, which is what you would expect. So I think those two pieces of evidence are pretty, to me, are quite convincing that this is a D1 specific mechanism. But it's a really good question.
Thank you. Okay, I think we're done with questions of science, but now we can start with the yeah. Please share your email, Christian, so we can have you contact also Dr. Cree. So let's start with the recruitment part. Dr. Hershey, if you would like to share your screen.
It's tricky.
Okay.
Hey, did that work?
Yes, it does.
Excellent. Well, yeah, I'm really glad to be here and to talk to you guys today about our Neuroscience PhD program at WashU. Just a word of warning, I am working from home, as you might have guessed, and I have 2 dogs that like to announce the arrival of packages and mail. So we're going to try to see if we can get through this before Amazon does its fine job. The other thing is I want to let you know that I have 2 current graduate students at WashU joining us. They should be coming online maybe around 4, and then at the end we'll have time for questions, and they will be able to give you sort of a hands-on view of what it's like to be in our program. They are both graduates of the University of Puerto Rico, so they have some good sense of where you all are coming from. So I was asked to give a little bit of an overview of our Neuroscience PhD, possibly the MD PhD program, and the summer programs for undergrads. But I am the co-director of the PhD program, so that's what I know the most about. I can answer a couple of questions about the summer programs. And then one of our students that's coming online later is in the MD PhD program, so he can help answer questions about those issues in particular. So I have a co-director, Tim Holy, who's not on today, but together we try to keep the wheels on the bus and keep this wonderful program moving forward. So our Neuroscience PhD program has a couple of really great qualities that you may see in other programs as well, other universities. But what we have is a very strong administrative structure. It's called the Division of Biology and Biomedical Sciences, or DBBS. And this umbrella organization supports 12 different PhD programs. So we have a lot of sort of uniform administrative support that allows us, the co-directors, to really focus on the students themselves. It also provides a lot of opportunities for students from those 12 different programs to interact with each other and learn from each other. So that's a plus. We also have a very long tradition of excellence in neuroscience, many Nobel Prize winners, other kinds of Causally Award winner, things like that. And so we have a lot to be proud of. We often rank in the top 10 of neuroscience programs in the States. And then in the program itself, we have a real depth and breadth in the training that we provide. We have interdisciplinary pathways, tons of program faculty mentors that come from all over the university, over 30 basic and clinical departments, 2 campuses, all sorts of resources and cores and machines and equipment and everything. And we're in the process of building what's supposed to be the biggest neuroscience building in the Western Hemisphere that will be done in a couple of years and will be filled with wet lab neuroscientists from all across WashU. But I think one of the things I'm most proud of is the fact that we have such a strong, vibrant, collaborative, supportive, and growing neuroscience community. So I'll tell you a little bit more about our unique features. So I think, we have great faculty. We have a lot of faculty. They cover just an incredible breadth of activities in neuroscience, but they're also very approachable people. They're very generous with their time. They really believe in graduate education, and they care deeply about their students. And I think that's really a wonderful combination, to have such great excellence, but also kindness and generosity, and just, being very good human beings all the way around. We also take care to try to train our students in all aspects of becoming a neuroscientist, right? So there's lots of content you can learn, but there's also a lot of skills that go into being a good scientist and also being a good community member. And we try to explicitly train students in those things. And then all of this, the connected community that's collaborative, faculty that are approachable, and excellent training is embedded in an extraordinarily livable city. We have low cost of living, we have tons of things to do, and that makes for a very high quality of life, particularly for graduate students who don't have a lot of time and they don't pulling in the big bucks yet. So it really makes it a very very high quality of life for students. So a little bit more about the faculty. I said they cover a full range of neuroscience, from studying individual ions to studying human decision-making using neuroimaging. There's also a lot of translational neuroscience that's not captured in this bullet point, but a lot of disease-related neuroscience that goes on. We have lots of opportunities for our communities to get together. Students and faculty get to know each other through lots of different events, like students doing rotations in labs, classes. We have lots of meet and greets, or TGIFs, little get-togethers. Right now, those are few and far between because of the pandemic, but normally that gives a lot of opportunity for having casual conversations with different faculty. You might meet faculty through seminars, going up to talk to them after their talks, journal clubs, and other informal events. And faculty and staff are part of each student's both formal and informal mentorship teams. So we really believe in the team approach to mentorship, that it's not just one person that has all the answers, that you really need to interact with a full range of people that each give you, you know, a different view of things, and then you get to sort out what you really think is the right thing to do. So here's the basic training program that we offer, the basic curriculum and milestones. So in the first year, well, first of all, I have these things color-coded, so everything in blue is more of a skills-based experience. The purple is core neuroscience content. The black is extracurricular required things. And then the green are optional things that you can do to enrich your experience in the program. So the first semester you're doing first year you're doing lab rotations in order to pick your thesis lab. You're also taking the 2 core neuroscience content classes, cellular neuroscience and systems neuroscience. You're also taking some really important skills-based class, graduate research fundamentals. I'll talk a little bit about that in another slide. Coding and statistical thinking in the second semester, and a grant writing class. And altogether, these should prepare you, along with the rotations and journal clubs and other events, prepare you for taking your qualifying exams at the end of that first year, which takes the form of writing up your own ideas for some studies that you would do to address a question that needs addressing in the literature. And then you turn in the written part and you do an oral exam part for that. And throughout, we're giving you a lot of advice. So the program directors meet with you and give you a lot of advising. And then there's other opportunities to get advice. Second year, you've declared a thesis lab. You're thinking about what is your topic for your thesis, working towards forming a thesis committee by the end of that second year. You take a presentations and ethics course. We're offering now, starting this spring, what we're calling Nano Courses, which is sort of a very brief, intense, deep dive into a particular topic or perhaps a particular technique. Actually, Dr. Creed has taught a few of our few workshops for the neuroscience community on some of the technology and coding software that she's been involved in developing that have been very popular, and that could be considered kind of a nano course where you go in for a short period of time and really get up to speed on a particular topic or skill. We also require you to do 1 mentor teaching experience. So you're a TA, a teaching assistant for a class in the either undergrad or graduate campus graduate programs related to neuroscience, and you actually get real teaching experience there. You get to be involved in either running lab demonstrations or lecturing. And you also get some training from the teaching center at WashU related to skills of teaching. In your year 2, you might also select a pathway. I'll talk a little bit about that in a minute. And any additional courses that you feel you need to enhance your training and your preparation for your career choice beyond. After you propose your thesis in year 3, you're continuing with any pathways or certificates that you might be going after, but you're really focused heavily on your research towards defense. Along the way, we do a lot of career exploration activities. There's student groups, outreach activities, leadership roles, and community events. So a little bit more about the skills that we want you to start with. So in the first semester, you take a class called Graduate Research Fundamentals with all other first years from all of those 12 programs. And you get a good sort of first view of some of these essential skills like scientific reasoning, responsible conduct of research, writing, communication, career readiness, importantly, managing yourself, managing stress, working with other people, managing conflict, and important principles about diversity, equity, and inclusion. So we try to cover all of that in just that semester. So it's a very sort of a brief view of all of these things, but all of these things will continue to come up throughout your graduate career and beyond. And so it's good to start at the beginning. It also gives you a way to get to know students across all of those programs, which can be useful in terms of just having a nice community around you and potential collaborations in the future. So some of the optional things that you can do, I alluded to already, the pathways. We have these very specific pathways around different interdisciplinary ideas. Some of the most popular ones for our neuroscience PhD students are the cognitive, computational, and systems neuroscience pathway, where you take a couple of extra classes beyond the basic requirements for the neuroscience program, and you're in this pathway with students from the Psychological and Brain Sciences PhD program and the Biomedical Engineering program. And it helps to form connections between those three disciplines and allow you to learn from each other and understand kind of the different perspectives of those domains. Another popular one is the Marquee Human Pathobiology program. This is a focus on translational neuroscience, going all the way from cells and molecules all the way up to a clinical syndrome. And you actually get to shadow a physician to see patients with whatever condition you're studying in that course. So that's very popular. And then there's also elective credentials that you can go for, depending on, you can use these as sort of career exploration activities or planning for, really building your resume towards a particular career, like science communication, entrepreneurship, teaching, leadership, and this higher education one. And so these aren't particularly intense, but there are some additional activities, maybe a class or some workshops, and then experiences and some sort of final product, like perhaps for science communication, it's something that you publish in a newspaper or other kind of publication around communicating science. So our community, as I said, is very strong. It's really one of the reasons why people stay at WashU for so long. I've been at WashU my whole career, and it's really hard to think about leaving the people. Like, you can find resources everywhere, or not everywhere, but lots of places, but it's really hard to find people that you find to be great collaborators, that make work fun, that make science fun, and make your science better. And so I just show a couple of pictures of a recent Zoom call we had to celebrate a faculty member's retirement. We had alumni calling in from all over the world, and just had a really great time. Current students, former students of his, faculty that have come and gone, and everybody gathered on the Zoom call, and it was really fun. Of course, it would have been a lot more fun in person, but we do what we can. And so another fun event is that Sally Vogt, our program coordinator, holds an annual picnic for students in programs that she coordinates, and over to her house. And so students always have a lot of fun there. That's this picture. And then we have an annual neuroscience retreat at Trout Lodge, which is in sort of south of St. Louis, in a very beautiful part of the state. It has a lake and hiking and things like that. We all go there for 2 days and hang out and listen to science and have poster sessions, and there's music and all sorts of fun things. So that's been a really very popular event. It brings together the entire neuroscience community, not just neuroscience program students. And a few other things I list there on the side that we do. And this community, is so strong and vibrant and friendly, and that makes life, a lot of fun, and going to work very fun to see your friends. But it also promotes really excellent science. So this is an example of a paper, and I could name many other papers that have these same qualities, where there are students and faculty from different programs, both neuroscience and in this case, immunology, faculty from neuroscience and immunology, and they pulled together sort of top-notch science to examine a question that's really at the intersection of these two domains. And it was just a really, oftentimes these are student-driven, right? So through interacting with your fellow students across programs, you might realize that together you could do something really cool. Then those students go tell their PIs and say, hey, we have this idea, and the PIs are like, yes, great, sounds wonderful, and something like this happens. So that's really fun to see. So I want to take a little bit of time to talk about our efforts around diversity in the neuroscience program and across all of the PhD programs at WashU. We have some really excellent staff, Joel Dalton and Rosie Jones, that handle anything related to diversity, equity, inclusion for students and alumni, and they're really phenomenal. Joel is just doing amazing things at WashU. And he's also a very kind person that students go to for advice. So here's one of your sort of an example of a staff mentor that students can go to. We also have a formal program called the Initiative for Maximizing Student Development. This is run by Jim Skeeth and Tracy Hermanstein. And this is a formal program that brings students together from underrepresented groups and supports them throughout their graduate career with some additional activities and workshops, and provides a really strong community for those students. We also have an Office of Neuroscience Research that has been very active in race, equity, and social justice in neuroscience. You know, it was always there, but I think this past summer really increased everyone's interest and sort of the urgency of these activities. And now we have a student-led journal club that's going to be starting this month, focused on these issues, where students will take turns talking about issues or papers that relate to this, these issues. And so we look forward to that. There's also lots of student activities that relate to diversity, and some that relate to sort of professional interests or outreach and advocacy, and these are all listed here. So these are all very vibrant programs that neuroscience students have often been involved with or led. A great way to get leadership skills as well. A lot of professional interest groups that really span from education to entrepreneurship, biomedicine, and writing or editing. And then we have some great outreach programs, the Young Scientist Program, which was started by PhD and MD PhD students, and has been going for now 20 or 30 years, I think. Very, you know, taking science type skills into classrooms, but we also have some that are specific to neuroscience, particularly the brain discovery, which works with, I think, elementary age kids. But then there's also other events, and I think one principle here is that students can decide what is important and where they want to put their time and passion, and get the support from the university and the program and the division to make these things happen that can have a really big impact on all the students that come later, but also an impact on the community. So we definitely value the student-driven activities. What are our outcomes like from our program? Average time to degree is 5.7 years. The national average is 6.9 years. For MD PhD students, it's a little shorter. It's more like 4.7 years. These show where our students go. This is across all those PhD programs. So it's not specific to neuroscience, but our neuroscience numbers look about the same, where we have a large portion. We might have a higher portion in academia, actually, for the neuroscience program. But you see people going into lots of other career pathways as well. And we support that. We provide opportunities for gaining those skills as well. Okay, what are the nuts and bolts here? So our financial situation for the neuroscience program, we provide you a $30,500 annual stipend. And then all of these other things are covered. So your annual tuition is covered, medical, dental, disability insurance is covered, and network access fee is covered. And then you can get a $600 check to use towards travel to meetings after you have proposed your thesis. You also get a free Metro pass. So you can go anywhere on the buses and the Metro in St. Louis, which is quite handy because the undergraduate or Danforth campus and the medical campus are 3 miles apart, separated by Forest Park, and the Metro runs right back in between those. So, so that's really a nice thing for students to go back and forth. Okay, what about St. Louis? First of all, it's right here. You're maybe down here. So a little ways away, but very nicely situated, close to Chicago. Has an airport and everything, and we have a very nice sized city that's extremely livable. So as I alluded, it's affordable. So I was a graduate student in St. Louis. I saved enough money to buy a car. So I think that's pretty good. And I didn't make $30,000 because that was a while ago. There's also tons to do, and a lot of it is free. So you can do just about anything you want with this salary and with the access you have. So St. Louis actually has more free world-class attractions than any place in the nation outside of Washington, D.C. I didn't actually know that till I put this slide together, but I'm not surprised. Also, there's a lot of things walkable just from the medical campus. We have a huge, I think it's 1,400-acre park called Forest Park that is adjacent to the medical center and in between the medical center and the undergraduate or Danforth campus, where actually some graduate students do work in labs over there. And Forest Park is gorgeous. We also have the Central West End, which is a really thriving part of the city with lots of restaurants, etc. The Grove, these are all walkable, and a lot of students live in these areas. Within Forest Park, you have the Art Museum, the Zoo, the History Museum, and the Science Center, and those are all free to enter, which is just remarkable. I think you can't find a city that has world-class amenities like this that are free. What I love about St. Louis, there's so many unique little nooks and crannies, so many cute, interesting neighborhoods with different history. They have lots of parks embedded in them, lots of restaurants, shop, live music all over the place, and beautiful architecture. And then if you don't like the city, you can go very quickly to the country. So we have the Ozarks area, which is southern Missouri, lots of beautiful, pristine rivers for canoeing, lots of climbing and canoeing, camping and hiking opportunities. They're very easy, and that's a favorite for our students. So now we have finished the presentation part. This is a very busy slide that I will, you will definitely have access to all of these slides, so you won't have to copy any of this down, but there's lots of great places to go and check us out online. In addition, I want to highlight that we are doing the SFN, Society for Neuroscience Graduate School Fair, starting tomorrow and going for 3 days from 11 to 2 Central Standard Time. So if you're interested in that, you can find that online and register. Come talk to us and the students. And then we're going to do some Just WashU specific open houses on Zoom on 2 Wednesdays in November. If you want to sign up and come, we'd be happy to talk to you there too. So again, this is me and Tim, the program directors. This is Sally Vogt, who's really the heart of the whole program and makes everything happen, and is a wonderful friend to all the students. And then I'm hoping we have Maria Gonzalez and Jose here online. Yes, I see Maria. Good.
Hey.
Okay.
Hey.
Thank you for, okay, so you're here. This is great. Jose, are you here? Not yet. Okay. Well, maybe he will, maybe he had a little trouble getting online. But anyway, Maria is a current neuroscience PhD student in her first year. And she is a graduate of the University of Puerto Rico and an Endure Scholar. And very happy to have her here to talk a little bit about her experiences. So Maria, maybe you want to start by saying a little bit about yourself, and then people can ask questions.
Okay. So hi everyone. I know this is a stressful day, so I guess thanks for joining this. I am originally from Arroyo, Puerto Rico, and I did my undergrad in the University of Puerto Rico at Calle. I think early on, as an undergrad, I thought that I wanted to go into medicine. And so how they teach us in the island, as you may know, is that if you want to go into medicine, you should go into research and kind of strengthen your CV. And so that's how I got into a summer internship and did research with Jacqueline Flores Otero at the Institute of Neurobiology. And so after that, I really discovered that I loved research and that I wanted to stay more in this area. And so I think as a second year as an undergrad, I applied to the BP program here at WashU, and I got admitted. And so for 3 summers, I worked with Josh Rubin doing sex differences in glioblastoma, which is brain cancer. And so from that point on, I just kept doing research. And then when I got to senior year of undergrad, I applied to graduate programs, and I didn't got into any of those. So I decided to apply to a prep program or post-bac at UNC Chapel Hill. And then I worked with Graham doing more sleep and more neuroscience research. And so after that, during the prep program, I decided to apply to grad school. And this time, the outcome was much better, and I got into the programs that I wanted to. And I was really familiar with WashU, and I knew that I loved the environment here. It's also one of the best neuroscience programs in the United States and in the world. And so when I was choosing grad school, I think one of the main reasons I came here was the community, and also the vast amount of research that you can do in so many different areas. So I kind of knew that I wanted to go into circadian rhythms. And there's so much people doing that here from such different perspectives that I just decided to come here. And I have been having a really good time so far, even when everything is through Zoom and virtual. And I've been able to like rotate in person. So that's nice.
Great. Well, thank you, Maria. That's a wonderful story, and I am glad that you are happy here despite everything being so strange for your first year. Well, great. We're happy to take any questions. And Meaghan, if you have anything to say about the program, feel free to join in. Or if anyone has questions, they can chime in or put it in the chat. I guess I'll stop sharing so that we can see each other.
I think one of the things that really jumped, so I was faculty on the East Coast before moving here about 2 years ago, and just what really jumped out at me was exactly what Tammy said and what Maria has said, that the community is really special, and just how quickly it's been to integrate and make friends. And when I moved labs, I had my technician at the time move with me, who since, she loved it so much, she also matriculated, and she's now a second-year PhD student. So it really does, it's really something it's hard to describe just how welcoming and how friendly people are, and how that leads to higher quality science, because people are just so open and collaborative. And at the student level, I'm already seeing a lot of cross-pollination, just how close-knit the graduate cohorts are, and the ideas that they come back from chatting with their friends in other labs and getting perspectives. So that's something really special that it's hard to put into words if you're not experiencing it, but I'll try.
Okay. Any questions for Dr. Hershey or Dr. Creed or Maria from the students? You can unmute yourself. We still have 5 more minutes. Any questions?
I'll also quickly say that aerial picture of the park that Dr. Hershey showed is, it's not just, this is a nice park in St. Louis. That's literally outside my office. That's the view from, the reverse side is the view from my office. And we have especially in COVID times, that's where my lab goes for lunches all together. So they can be outside and be distant. So it's not something abstract that, oh, there's a park in the city. This is where they spend their weekends and their lunch hours, which is, again, pretty special.
Any questions?
Could you give us a little insight on undergrad opportunities, research opportunities? I know you said you're from the PhD program, but if you know anything.
Yeah, so we do have 2 very strong undergrad opportunities for the summer. One is the Amgen program, the Amgen Scholars Program, and the other is called Biomed Wrap. And I have information on those 2 programs in my slides. So can I send the slides?
Yeah.
Will that work?
You can share them with us. And remember, this is taped, so the students can access at our website as well. So.
Okay, Wonderful. So yeah, the two programs you apply to, and there is a stipend, and they help you, get settled in St. Louis and find a lab. And then there's other kinds of structured activities besides being in the lab to enhance your experience there. And then of course the Endure program, a lot of students choose WashU labs for those experiences.
Great, thank you.
You're welcome.
Any other questions?
I could just say that MD PhD program, if anybody's interested in that, is a very competitive program, but we do get, so people apply to that program and then they do 2 years of medical school and then they choose which PhD program they want to go into. So every fall, we get students from that program joining the PhD program in neuroscience. So every year it's maybe 5 or sometimes more of those students, and they're just wonderful students and incredibly well-rounded, and the MD PhD program is extremely well run. They're very well taken care of. So I think if you want more details on that, you'll get a lot better information if you go just to their website and contact them directly.
Okay. Any other questions? Okay. Well, I
had a very silly point. Sorry.
No, go ahead.
Sorry, speaking of this is ridiculously silly, but I noticed I'm using my husband's computer because the backdrop is nicer, and I'm realizing that I have his name under my face. So I was wondering if you would mind maybe just sending out my final slide of my presentation with my contact information and my proper name to everybody in case you want to know more or get in touch.
Sure.
As we said,
And
a little
her
grace.
husband is a neuroscientist, so it's sort of,
Yes.
I
know, but he already soaks up all the BP and DER students, so I don't need to give any more traffic his direction. So it's actually Maya Blewett was in his lab, but we have a joint lab, so she's he's mentored BP and DER students as well. So
Okay. Wonderful. So I guess we have come to the end. I know if there are no other questions for our invited speakers, and I wanted to thank both Dr. Meaghan Creed and Dr. Tamara Hershey and Maria for being here and talking about the WashU endearment for the University of Washington School of Medicine, which is a wonderful, wonderful institution, which has made a considerable effort of improving not only the future neuroscientists' careers, but also providing them with the environment where they can excel. So I really, really I'm very impressed, and in addition to that, thankful that you were able to come to our BP Endure seminar series. Here in the UPR, you're always welcome, even though we're doing this virtually, but anytime you guys want to come back, just let us know, and we'll certainly have space for you to talk to our most wonderful students.
They are. I had Maria Santana Rivera in my lab last summer, so they're not 2 summers ago. Yeah, so she's wonderful.
Thank you.
Yeah. Well, thank you for inviting us. We really appreciate it.
Thank you.
Of course. And so join us next week when we will have Dr. Jolan Smith from Emory University. He's going to be talking about thalamic pathology in Parkinson's disease, a potential source of early cognitive impairments. So I hope to see everyone next time, and have a great rest of the week. Thank you. Bye.
Bye.
Thank you.